Hello and welcome everyone. In these next few podcasts for epigenetics, we're going to talk about DNA methylation. Let's think about what we've talked about the past few classes. We've talked a lot about this here, DNA and the histones around it forming the nucleosome, and how this nucleosome would help hide, so to speak, a gene that might be in here, so it wouldn't be transcribed. We talked about the importance of forming these nucleosomes in establishing that higher order structure. We then recently talked about how in order to destabilize this nucleosome, we could pull some of the DNA out. What I want to focus on today is how we're going to methylate this DNA and the importance of methylating the DNA to help determine which genes will be turned on or turned off. So we're going to begin by talking about some of the basics of DNA methylation, what it is, where we see it, and in this discussion we're going to talk a lot about CPG repeats or islands of CPG repeats. Then we're going to talk about 4 different mechanisms of how methylation of the DNA affects transcription. How does the presence or absence of these methyl groups here determine whether or not this gene is going to be expressed? Then we want to talk about the enzymes, which we're going to call DNMT, how these enzymes transfer methyl groups here, some of the properties of these enzymes. And then we want to close with regulating DNA methyltransferases. And as we discuss this topic later on, we'll see this is very important because we don't want to turn on all the genes or turn off all the genes. It's going to be important that we regulate when and where DNA methyltransferases operate. But we're going to begin talking about DNA methylation, just some of the basics of it. So let's go ahead and get started. We're going to spend quite a bit of time talking about DNA methylation, but what I want to do in this podcast is just to introduce this concept of DNA methylation and some of the larger pictures of how it's affecting gene expression. Now remember, we have 4 common bases that are found in DNA: cytosine, guanine, thymine, and adenine. There's also uracil that's found in RNA, but I'm just talking about DNA for now. There are variations of some of these bases, and one variation that we want to talk about is 5-methyl cytosine. This occurs due to an enzyme called DNA methyltransferase. And all that does is add a methyl group to the 5-carbon position of cytosine. So usually cytosine looks something like this. It's a single ring, 2 ends on these corners here, carboxyl group. And when it's methylated, it looks the exact same with one small exception, but with big impact. And it has this methyl group here on the 5' carbon. And this methyl group, as we'll see, even though it's a small structure, it makes a huge difference in the ability of genes to be transcribed. I should mention that the reverse can also happen, where you take 5-methylcytosine and create cytosine, and that's done using DNA demethylase. So what does it do? So 5-methylcytosine has for quite some time been proposed to decrease gene expression. However, we don't necessarily believe this is a universal mechanism, because in species like yeast and fruit flies, there's very little detectable methylation of the cytosine. So this is some evidence saying, well, maybe it's not as important as some people think. Though in mammalian models, like mice, we know that when you prevent the methylation of cytosines by mutating DNA methyltransferase, so mice with a knockout of DNA methyltransferase results in developmental defects, and in some cases, embryonic lethality. So at least for mammals and invertebrates as well, methylation does appear to be important. In fact, essential in some cases. So the question might come up is, did mice just somehow or another develop this DNA methyltransferase? How early of an evolutionary development of this enzyme was there? Well, we know that there are methyltransferase genes are found in bacteria. So it's not a new enzyme. This enzyme's been around for a long time. It has slightly different targets. It can, in some species, it can methylate other nucleotides. As I said before, we don't see a lot of methylation in yeast and fruit flies, but they do have methyltransferase genes. It's just that they don't methylate the cytosines. So why don't I put that, of cytosines? So when it exactly occurred is debated, and there's some phylogenetic studies that are being done to try to trace it back. And I don't want to spend time talking about that right now, but what I would like to talk about is maybe a possible why. Why did it develop? Did it develop in an early eukaryote or an early vertebrate, or maybe in a plant, to control expression of genes? Perhaps. But a prevailing idea is that DNA methyltransferases, which we abbreviate as DNMT, and there's several of these, but that they may have arose at some point during evolution to fight off viruses, just to survive. And through the process of natural selection, maybe some of these DNMTs began to preferentially methylate certain genes in an early eukaryotic cell to help regulate the timing of expression. How could it fight off a virus? Well, some evidence for this is that some viruses, not all viruses, they have these things called CPG repeats, or sometimes we call them CPG islands. It's probably a better word to use here, where there's a lot of cytosine guanine repeats. We're going to talk about CPG islands in just a moment, but what you should know for now is that these CPG repeats are methylated. So you could imagine a system where a virus has this CPG repeat in its genome. When it integrates into its host genome, it gets methylated. Those genes now are not expressed, which could prevent the virus from completing its cycle. If this is what's happened, we don't know. We just have evidence from viruses, what they look like. We're beginning to do phylogenetic studies so we can trace back when the first DNMTs arose. And over time, we will be able to piece this together. But it's interesting to think, if it turns out to be true, that the reason we have methylation and this rather advanced ability to regulate our genes may have really started off because of an early ancestor's need to fight off a virus. So that's a nice segue into CPG repeats. So let's talk about those. What is a CPG repeat? We've already mentioned what they were on the last board, but these are cytosines next to guanines. And when we call them CPG islands, what we're saying is there's a whole string of them, a CG, 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 several in a row. I also want to be clear when I say CPG repeats, what I'm talking about is the linear sequence. So you'll have a cytosine, a guanine, a cytosine, a guanine, a cytosine, a guanine, and so forth. I do not mean the base pair of C to G, C to G, C to G. I don't mean that. I mean a linear sequence like we see here. So not this. Okay, now that we just introduced these CPG islands, I want to talk about two broad classes of genes that would have these CPG islands. The first are genes that have a higher concentration of CPG repeats than would be predicted. Different people define this a little differently, but in general, if you have a stretch of DNA of greater than 200 base pairs, and in that stretch, roughly 50% of those base pairs are the CG repeats. These are areas that we say have CPG islands, large stretches of cytosine and guanines. Because of all the CGs in these regions. As you scan a genome by looking at the sequence or staining it, these areas are very easy to identify. Since cytosines in these repeats is methylated, it was surprising to learn that cytosines in large CPG islands are usually not methylated. There are, of course, exceptions. There's this gene called MAGE, M-A-G-E, where they're methylated in somatic cells. This gene, MAGE, but not methylated in germ cells. But this is a rare exception. We also see some of these CPG islands methylated in cancer tissue, which may help explain some of the aberrant gene expression in cancer cells. The second class are genes with low CPG content. And by low, I mean about 1 per 100 base pairs would have these CPG repeats. This is usually seen in tissue-specific genes. Those genes that are expressed in one tissue but not the other tissues. You might also see these in genes that are temporally regulated. This first one is definitely seen in the tissue-specific genes. The temporally regulated one is more hypothesized because of the differences in the expression. Now, if you remember on the last board, I said those genes with a lot of CPG content are not methylated. So again, low CPG content are typically associated with tissue-specific genes and temporally regulated genes. A good example of this temporal regulation revolves around these two genes, OCT4 and nano-G. If I were to draw a graph here, I would put expression here and time of development on the horizontal axis. And what we see is very high expression early in the embryo during development. And as those ectoderms and endoderm layers begin to differentiate, the expression goes down and stays down for the rest of the adult life. And what we see is methylation, is the genes being methylated during this time. So if we were to draw another axis over here and call this methylation, we would see low methylation early on of these two genes that would slowly increase over time. So it's proportional, this expression, with the methylation during development. Another example is that of the gene called TERT. It encodes a reverse transcriptase that's important in maintaining telomeres, the ends of chromosomes. Now, these ends of chromosomes, just to remind you, are sequences that help stabilize the very ends of the chromosomes, so they stay a constant length and they don't begin to destabilize. Telomeres are very important in the embryo, and so TERT is expressed in the embryo. So I'm just going to write here, TERT is expressed with an up arrow. It is less important in somatic cells of the adult. All right, TERT would be predicted to be down-regulated, not expressed. And then we also see it in stem cells. In stem cells, TERT is expressed. It is also expressed in cancer cells. So, in the embryo, when TERT is expressed, methylation is lower. In somatic adult cells, methylation is higher because the TERT gene is not expressed. Stem cells and cancer cells, methylation is decreased. Okay, that ends our first podcast on DNA methylation. In the next podcast, you'll see that we're going to talk about the mechanisms of how DNA methylation affects transcription. If you have any questions about the material in this podcast, please make sure you come see me. If not, I'll see you in class. Bye.